Welcome to the Flames Lab. Flames Lab stands for Field Laboratory for the Assessment of Multiple Ecological Stressors. Welcome. This is a lab that tries to prevent any kind of contamination. So the first thing we have to do is put on our booties and step on the sticky mat to prevent anything coming from the outside. Thank you. So this laboratory is divided in various areas. This is the office area here, where we have our computers, where we store our data. And this is the microscope area, and also the area where we quarantine the animals. This laboratory is an ecotoxicological laboratory working with small lake animals called Cladocera, specifically Daphnia. So we take our samples here, we quarantine the animals here, we don't have any quarantines now, and then when we are sure that they are healthy from the field, they can go into the laboratory. In this laboratory, we also have a preparation area where we bring the samples that have been quarantined and then are healthy to put in specific jars. We have, because we have a chemistry area, we also have this hood where we can keep the chemicals that need to be in the hood. For example, acids or metals, they have to go there. We also have 8 refrigerators where we keep our media. We have different culture media for different specific experiments. So we have flames medium, we have combo, and we have bulk basal medium, and we also have medium for diatoms. So these are our stocks, and from here we prepare the culture media. These media have also vitamins that were specifically designed by other scientists, Dr. Michael Lynch, and we have to keep them in the freezer and add it to the medium to have a complete nutrition of it along with the algae. So come here. This is where we keep our food. Thank you. This is where we keep our food. So each one of these tubes contains algae that have been growing in the bulk basal medium. And when we want to feed the animals, we resuspend the algae and we administer a certain amount of cells per liter. So the animals are well fed and can grow, have lots of eggs and lots of babies. This is the chemistry area where we prepare the medium. So here we have very basic activities for chemistry. We have a balance that we use to weigh the reagents, and we keep all our reagents here separated from the animal area, and we produce here any formulas that we need for that. We come to the heart of the laboratory. The heart of the laboratory is the chamber room. These chambers have been here to help us control temperature and light regimes for the animals and for the experiments that we run. So for example, here we have the Daphne collection. And this chamber let us have the animals here with a day-night regime. So this means that these chambers, the lights go off at around 8 p.m. and then they come up at around 6 a.m. So the animals have this daylight regime, kind of a summery light regime, and the temperature is always at 20 Celsius. Why do we want them at 20 Celsius? Because it is the best temperature for them regarding reproduction and regarding survival, and also for the food, for the algae that they are feeding. So all the laboratory is at 20 Celsius. Also, because the animals are at 20 Celsius and the media have to be, the media that are there in those cardboards, They are also at 20 to prevent shock for the animals, temperature shock for the animals. So these daphnids have allowed us to do multiple experiments with metals. We have worked with copper and nickel and chromium, and now we are working with sodium chloride, determining the levels of chloride that are allowed or that are convenient for the lakes here in the central and northern parts of Ontario. We determined in this laboratory the point of recovery for the Sudbury lakes that were historically impacted by copper and nickel. And when we found it, it was very exciting because that's when the animals, when we found the limits, also the animals were present in the lake. So it was very exciting to have this parallel of nature recovering and testing in the laboratory for that. We also did some experiments with chromium-6 regarding the potential for chromium exploitation and exploration in the Ring of Fire. And also because it is crucial in these lakes to have enough calcium to get the animals surviving, because these animals are crustaceans, and there are other crustaceans, many crustaceans in the lakes, microcrustaceans, and they need a certain amount of calcium in order to survive. So they need them for metabolic functions, but also for their exoskeletons. And we have been noticing in the Dorset Science Center that the calcium decline in the lakes is going to dangerous levels. So if the calcium declines, the animals are not going to have enough nutrients, or this nutrient, in order to do metabolic functions and produce their exoskeletons. When they lack the calcium, also the neonates or the babies that they're going to have can be affected. So these studies have been done in this laboratory. And we also noticed that the calcium and the sodium interact with metals in the environment. Metals in the environment are everywhere. Some of them are nutrients, some of them are contaminants that are not nutrients, like chromium. So we studied the effect of this balance between calcium, sodium, and copper and nickel, and we found that both have an ameliorating effect regarding the presence of the metals. So when there is more calcium and more sodium, the metals are less toxic. But because we live in this area of Muskoka, in central and northern Ontario, the lakes are very soft. That puts a higher risk for the animals regarding this contamination. And then, because we found this interesting point with the sodium, we started testing sodium chloride. Why do we test sodium chloride? Because the winter maintenance of roads has an important excuse me, component in sodium chloride. The salt, the road salt that is added for winter maintenance, finally ends up in the aquifers and in the lakes. There is a guideline for chloride in the lakes from the government, from the Ontario government. But we found that excuse me, we found that because the water is so soft, the chloride levels that are in the guideline are too high for the survival of this Cladocera. So then one of our current experiments is related to chloride limits for saltwater lakes, or the animals in saltwater lakes. Why are these daphnids important, you can ask? Because they are so tiny, and they are so, just a microscopic animal. I mean, you can see it like in a petri dish or in a jar, but they are really tiny. They are crucial animals in the lakes because they are a very important component of the food chain in the lakes, and they also clean the lakes because they eat so much. They eat algae. So when we have a healthy population of Cladocera or these daphnids, the water is going to be cleaner from algae because that's what they use for their food. And then there is going to be a lot of daphnids, and that is very good for the fish because they are food for the fish. So in this little, part of the food chain, we find really a treasure, ecologically speaking. And here in this area of Muskoka, we want to keep our lakes clean and healthy. So one of the things that is increasing is the chloride in the lakes because of this road winter maintenance that I was talking about. So we have been studying that, but we also know that there is interest in finding a substitute for the road salt. And one of those substitutes is beet juice. Yes, beet juice, like the beet juice that we drink in the morning, sometimes. That beet juice can be added to the roads and then can substitute the road salt and expectedly have a lesser impact on the water. We are doing that research, and the findings are very interesting because sometimes the good thing might not be very good in other aspects. So maybe the chloride can be reduced, but maybe the organic matter is increased, and the organic matter is never good to go into our lakes because then that produces more production of algae, and then we go back to the same problem. Also, the daphnids have a certain potential to clean the water, but if it is increased by other nutrients, then it becomes very difficult, and the food chain starts changing. So all our work here is for trying to explain the monitoring trends that are found by the ministry, explaining how the trends affect the daphnids that are these crucial indicators in the lakes. They're indicators because they are very sensitive to any change. So any change in anything can affect their population. So they are excellent indicators. So then we have been trying to explain this calcium decline and the chloride increase and the metal impacts and the metal amounts in the lakes using these indicators. We have also been doing, not me particularly, but we have also been doing invaders. We have worked with Bithothrappus, which is this spiny water flea that is a disaster in the lakes. This invader eats the daphnids, the baby daphnids, and is very bad for small fish, for the larval fish, because it has a spine, and then it affects the digestive tract of the fish. So in the past, Dr. Yan and Dr. Forget her name. A couple of other scientists have been studying those effects of the spiny water flea. There have been many students, graduate students, postgraduate students working in this laboratory. That is truly a gem to Muskoka, because we are exploring how to protect the water in Muskoka, central and northern Ontario. It is not only good to have a lot of water, that we do, but it is important to have good quality of water. And to have good quality of water, we need to make sure the system, the ecosystem of the lakes works fine, works in these pristine conditions as we have them. So this laboratory has been doing this important work for the past 17 years or more, probably 18 years. And well, we are very proud of what we do, and we hope that we can keep serving not only the ministry that we produce data for biomonitoring and for policy right now, for the policy area of the Ministry of Environment, which we are very happy to be helpful, but also for the citizens of Ontario, for the people who owns the cottages around here and want to have always their nice, clean lakes. That impacts not only the pleasure of being in nature, and being in the water, but also the value of their properties. So all these things come together, and this laboratory is just a little part of this social environment that keeps growing in this area, that keeps so clean in this area. And this laboratory with the scientists, with the science that we do, is trying to support and to protect all the nature in the region for everybody, even for the animals, of course. I'm Dr. Martha Celis, and I am the researcher here in the laboratory. I manage the laboratory, and I have been doing the research that I just told you about. Thank you.